The last category of cell organelles slash cellular structures is this hodgepodge category of any components that participate in structural support of the cell, movement, and then communication, usually between cells. One of the most important parts of this particular category of cellular structures is the cytoskeleton of the cell. So again, cyto stands for cell, and then skeleton is for skeleton. Unlike your skeleton, which is essentially made out of bones, right, which are made out of various minerals along with cells, the cell cytoskeleton is actually a network of protein fibers that's organized within the inside of the cell throughout the cytoplasm. And the cytoskeleton itself has a variety of different functions depending on which of the fibers you're actually looking at. Just to give you sort of a preview of some of the functions of the cytoskeleton that we're going to be looking at. So first of all, it supports the cell's structure. It organizes many of its activities. Sometimes it anchors organelles in place. In other cases, it might actually end up moving the organelles or even maybe moving the cell itself. So let's take a look at the three different kinds of fibers that make up the cell cytoskeleton. And they're listed down below on this particular slide. So we've got our microtubules. These are going to be the largest of the three types of fibers. Then next we have the microfilaments. These are going to be the thinnest. And then the intermediate filaments, as the name implies, are going to be somewhere in between the microtubules and the microfilaments in terms of their diameter. So they're intermediates. So the picture down at the very bottom is a fluorescence microscope image of a cell, and in this case we've used three different fluorescent markers to label three different things within the cell. So first of all, you've got your nucleus in blue, right? That's going to house all of the DNA. And then you have two other markers labeling different fibers of the cytoskeleton. So you've got the green fibers that are sort of radiating from the center of the cell outwards. And then you've got the red fibers, which sort of encircle the perimeter of the cell. So let's take a closer look at the microtubules first of all. So the microtubules, these again are going to be the thickest of the three different kinds of fibers we're going to look at that make up the cytoskeleton. These guys are made out of a protein that's called tubulin. Now, tubulin comes in essentially two different flavors. So there's alpha tubulin and then there's beta tubulin. And one of each kind of tubulin come together to form a dimer. And then those dimers are arranged sort of in a helical shape to create that tube-like fiber. Hence why we call these fibers the microtubules. Functions of the microtubules include shaping the cell. They also help to guide the movement of organelles, or sometimes even the cell itself, if the cell has the capability to move. And then a really important function that we're going to take a look at later in the semester when we look at cell division, the microtubules are actually responsible for pulling apart the copies of chromosomes as a cell prepares to go through division, right? So when a cell does divide, one cell is going to split into two daughter cells. And when this happens, the two daughter cells, they need their correct genetic information in order to be able to function the same way as the original parent cell. So each cell needs its own copy of the chromosomes. So in preparation for division, the cell is going to it's going to multiply all of its DNA, so make an exact copy of the DNA, and then it's going to divide those two copies of the DNA, give one set of chromosomes to one cell and another set to the other daughter cell. And again, it's these microtubules that help to separate those two copies of the chromosomes. So let's take a closer look at this second function of the microtubules first, right? So this second function is guiding the movement of organelles, or also the cell itself. So you, as a human being, can move, right? There are two systems that help you in moving your entire body. So you have your skeletal system, your bones, and your bones can't move without functioning in conjunction with your muscular system, so your muscles. So a cell, if it's going to move or it's going to move materials within the inside of the cell, it needs kind of both of those halves as well. So it needs the skeletal system and it needs the muscular system. Now, the skeletal system in a cell is going to be the cytoskeleton, or these fibers that stretch throughout the inside of the cell. And then the muscles are going to be another kind of protein, which we talked about earlier in chapter 5. And these proteins are going to be the motor proteins, and they function kind of like the muscles, right? So putting the two together, a cell is able then to move various components on the inside. So much like you see in the lower right-hand corner here, this is a scanning electron micrograph of a squid giant axon showing a long microtubule, part of the cytoskeleton, which is functioning kind of like a track. And this track now is used by a motor protein. So if you take a look on the left-hand side here, on this little bottom portion here, what looks like the bottom half of a stick figure, right? So you got the torso, and then you have, so the torso there, and then the two little legs. That's the motor protein. And the motor protein is literally going to walk down that microtubule while carrying a vesicle, much like what you see in this video here up at the top. So you can see all those fibrous tracks and the little organelles and moving down that particular track. All right, so this is a way for cells to move various substances on the inside of the cell. Another way the microtubules participate in movement is actually moving the cell itself. So if a cell has capabilities to move, it'll have typically one of two different structures. Either it'll have a flagellum, or a flagella if it's plural, or it'll have a cilium, or cilia, plural. And these two structures are actually very similar in terms of the fact that they have these microtubules on the inside of these long appendages that are attached to the outside of the cells, and they're essentially just covered then in plasma membrane. So similarities there in terms of structure, but the flagella are different from the cilia. And so let's take a look at some differences. First of all, if a cell has a flagellum, typically, this is true for eukaryotic cells, the cell is only going to have one flagellum. And the flagellum itself has a very specific kind of motion. So the microtubules are bent on the inside of the flagellum by the motor proteins. And so the flagella is going to move sort of side to side. It's going to undulate kind of like a snake, right? And in the process of moving back and forth like that, it'll actually move the cell itself forward. So this is an example of a series of sperm cells with those flagella, and they are allowing the flagella then to propel the sperm cell forward. Down below, this is a little video of a rodent sperm cell, and you can see how the tail there at the bottom is going to move back and forth, propelling the head of the sperm up at the top there through the solution. Now, another way that a cell can move is through the use of cilia. Cilia are going to be a lot shorter than the flagella, and also if a cell has cilia to move the cell, it's going to have a lot of them. It's essentially going to coat the entire outside of that cell. And down below is a video of a paramecium that's actually swimming through a solution. You can't see the cilia, but they coat the outside of that paramecium, helping to propel it through the pond water, for example, that it's swimming in. The motion of a cilium is also very different than a flagella. So instead of moving back and forth kind of like a snake, the cilia is going to have sort of a power stroke and then a recovery stroke, and then a power stroke and a recovery stroke. So think of you being in a rowboat, right? And you have the power stroke and then you recover, power stroke, and then you recover. Same idea for the cilia. So cilia and flagella on the inside, they both have these microtubules along with the motor proteins that help then to move either the cilium and the flagellum, and that will inadvertently help to propel the cell forward as well. One last place where the microtubules are important. So we talked about certain structures that plant cells have that animals don't, and vice versa at the beginning of this chapter. And we introduced this area, usually close to the nucleus, that's called the centrosome. And the centrosome, remember, is the microtubule organizing center. So, for example, in animal cells, if we were to draw, let's say, a cell, right? So here's plasma membrane, here's a nice little nucleus. The centrosome, again, has no definitive boundary, but it's an area close to the nucleus usually where these microtubules are going to be organized and built from. So as a cell, for example, is preparing to divide, it's going to send out all these really long microtubules from the centrosome in preparation for splitting up the chromosomes during cell division. On the inside of the centrosome, this microtubule organizing center, in animal cells, there are two very specific microtubule structures. And these guys are always sort of perpendicular to each other, and these guys are called centrioles. So if we go back to the PowerPoint, these guys right here are going to be the centrioles, and the hazy yellow background, that's the centrosome. So you'll see that the centrosomes, to me, they look like noodles, and they have a very specific organization. So they're going to be 3 short microtubules organized in a row, and then 9 of those triplets are sort of put together in a starburst pattern all the way around there. Forming one centriole, and then you can see that there's a second one here that's perpendicular to the first. Now, what the function of these guys is, we have no idea. Scientists have zapped them with lasers to eliminate them, and the cells can still go about and function perfectly fine, go through division with no problems. So potentially an evolutionary relic as far as we can tell. But again, these centrioles are made out of these microtubules, and they're found within the centrosome. All right, second fiber of the cytoskeleton. These guys are a lot thinner than the original microtubules. These are actually the thinnest out of the 3 types of fibers that make up the cytoskeleton. And these are called the microfilaments. The microfilaments are made out of a different kind of protein. This protein is called actin. And so the actin subunits down here below, each actin subunit is put together with a whole bunch of other actin subunits, and 2 of those actin sort of subunit necklaces are twisted together to form a complete actin microfilament, as you see here. Now these guys have a little bit of a different function from the microtubules. These guys tend to bear tension or pulling forces, and they're highly prevalent in muscle cells, for example, right? So your muscle cells are going to be contracting and relaxing, so there's a lot of pulling on those cells, and you don't want them to rip, right? So as the muscle is contracting and relaxing, you want to make sure that you maintain that structural integrity of the cell, and the microfilaments help to do that. The microfilaments also help with cellular movement. And going back to the muscle cell example, this is an image actually that we've seen before when we were looking at examples of different protein functions back in chapter 5. And one of the functions of proteins was the fact that they can function in contraction and as motor proteins. And so in your muscles, you actually have 2 different kinds of proteins that help with muscle contraction and relaxation. So in this particular image, we're looking at striated muscles. And the muscles are striated in this case because they have a very specific pattern as to how these different fibers or filaments are organized, right? So the actin filaments of the cytoskeleton in orange here are organized parallel to each other. And then in between them, we have a different kind of protein. These are your myosin filaments. And these guys have little heads on them. And the heads kind of play almost like tug of war. And they pull on the actin filaments, and then sort of allowing your muscle cells to contract, or they can relax and release those actin filaments, allowing your muscle cells then to extend. And this is essentially the basis of how your muscle cells and your muscle tissue ends up contracting and relaxing.